Somewhere in a limestone cave outside Beijing, sealed in darkness for 40,000 years, lay the skeleton of a man who would eventually force the entire scientific world to rewrite everything it thought it understood about who the people of East Asia actually are, where they came from, and whether the very concept of separate human races could survive contact with his DNA. When researchers finally extracted a usable genome from his remains in 2013, what they found inside those ancient bones did not simply add a new data point to the human story. It quietly dismantled a 200-year-old scientific and cultural assumption, one that had shaped textbooks, immigration policies, and popular belief across multiple continents, the assumption that humanity could be neatly sorted into a handful of biologically distinct races. This is the story of how scientists systematically compared the DNA of people across China against genetic samples collected from virtually every corner of the globe. And in doing so, uncovered a truth about human ancestry so intricate, so deeply interconnected, that it makes the old racial categories look less like science and more like an outdated map of a world that never actually existed. The skeleton that started this particular chapter of the story was discovered at a site called Tianyuan Cave, and the man it belonged to lived roughly 40,000 years ago, making his genome, at the time of its sequencing, one of the oldest modern human genomes ever successfully recovered from Asia. A team led by researchers from the Max Planck Institute for Evolutionary Anthropology in Germany, working in careful collaboration with Chinese paleoanthropologists, painstakingly extracted and sequenced his DNA using techniques originally pioneered for studying Neanderthal remains. What they found immediately upended assumptions about the timeline of human settlement in East Asia. The Tianyuan Man's genome revealed that he was already genetically related to many present-day Asian and Native American populations, meaning that the deep ancestral roots connecting modern Chinese populations to the rest of Asia, and eventually across the Bering Land, bridge into the Americas, were already firmly established 40,000 years ago, tens of thousands of years earlier than many researchers had previously assumed. It also showed something curious, that his lineage did not directly and simply lead to any single modern population, but instead represented an early branch of a much larger, more complex family tree, one with several branches that would go on to diversify, migrate, and occasionally mix back together over the tens of thousands of years that followed. To understand why this single skeleton mattered so much, you need to understand the scientific baggage it was quietly demolishing. For much of the 18th, 19th, and early 20th centuries, Western scientific institutions promoted the idea that humanity naturally divided into a small number of discrete races, categories often labeled using terms like Caucasoid, Mongoloid, and Negroid, each supposedly possessing distinct, biologically fixed characteristics, passed down cleanly through generations. This framework was used, disturbingly, to justify colonialism, forced labor, immigration restrictions, and outright atrocities across multiple continents, all under the false authority of supposed scientific fact. The term Mongoloid, in particular, was applied broadly and crudely to essentially all of East Asia, treating hundreds of millions of genetically and culturally diverse people, from Siberian reindeer herders to tropical rice farmers in southern China, as though they were a single, uniform biological unit. When the tools of modern genome sequencing finally became available, capable of directly reading the 3 billion letters of genetic code inside a human cell rather than relying on superficial physical measurements like skull shape or skin tone, scientists gained, for the first time in history, the ability to actually test whether these old racial categories held up against real biological data. The results, replicated study after study, population after population, have been consistent and unambiguous. They do not hold up, not even close. The clearest way to understand why comes from a concept population geneticists call clinal variation. Imagine standing at one end of a very long, very gradual color gradient, moving smoothly from deep blue at one end to bright yellow at the other, with every shade of green in between blending so gradually into its neighbor that it is genuinely impossible to draw a single clean line and say, here is where blue definitively ends and green definitively begins. Human genetic variation across the surface of the Earth behaves almost exactly like that gradient. As researchers sample populations moving gradually across geography, from northern China through Mongolia, across Siberia, or from southern China down through Vietnam and into the Malay Peninsula, genetic diversity shifts smoothly and continuously, with no natural objective boundary anywhere marking a supposed division between one race and another. When the massive Human Genome Diversity Project, and later the enormous Genome Asia 100K initiative, an ambitious collaborative effort specifically designed to fill critical gaps in Asian genetic data that earlier Western-led genome projects had largely overlooked, sequenced, and compared genomes from over 200 distinct ethnic populations across China and the wider Asian continent, the resulting data confirmed decisively that the amount of genetic variation found within a single traditionally defined racial category including populations broadly grouped together in China, is often larger than the average variation found between that group and neighboring populations elsewhere in Asia. In plain terms, two people from different regions within China can, in a genetic sense, be more different from each other than either one is from a person living in Korea, Vietnam, or Siberia. Race, as a rigid, discrete biological category, simply is not written into the genome. What is written there instead is something far more interesting, a continuous, branching, endlessly overlapping record of migration, isolation, and reunion stretching back tens of thousands of years. That record begins, like every human story eventually does, in Africa, and the journey from there to China unfolds along two broadly distinct ancient migration corridors that researchers have painstakingly reconstructed by comparing modern Chinese genomes against ancient DNA recovered from sites scattered across the entire Asian continent. The first wave appears to have followed a southern coastal route, moving from East Africa across the Arabian Peninsula, hugging the coastline of South Asia, and eventually reaching Southeast Asia and southern China tens of thousands of years ago. The second, somewhat later wave moved along a more northern inland route, crossing through Central Asia and eventually populating Siberia and northern China. Crucially, these were never two hermetically sealed, permanently separate populations. Genetic evidence gathered from ancient burial sites across China, including remains excavated at a site called Devil's Gate Cave in far northeastern China, dating back roughly 7,700 years, and comparative samples recovered from Neolithic farming. Settlements along both the Yellow River in the north and the Yangtze River further south reveal repeated cycles of these northern and southern lineages, meeting, mixing, separating again due to climate shifts or mountain barriers, and then mixing once more over the following tens of thousands of years, modern. Han Chinese populations, who today make up the overwhelming majority of China's population and represent, by sheer numbers, the single largest ethnic group on the entire planet, actually carry within their collective genome a layered blend of these ancient northern and southern lineages, with the specific ratio shifting gradually and measurably as you move geographically from Beijing in the north down toward Guangzhou and Fujian province in the south, exactly the kind of smooth clinal gradient that makes the old idea of a single uniform racial category scientifically meaningless. One of the most fascinating threads researchers pulled on involves a question that had puzzled linguists and archaeologists for generations. How did rice farming and its associated languages spread so explosively across an enormous swath of Asia and the Pacific? The answer turned out to be written directly in DNA. Ancient genomes recovered from early Neolithic farming sites along the lower Yangtze River, the birthplace of rice. Domestication roughly 9,000 years ago show a clear and traceable genetic legacy in modern populations, extending far beyond China's current borders, into Taiwan, and from there, remarkably, out across the vast expanse of the Pacific Ocean into the Philippines, Indonesia, Madagascar off the coast of Africa, and eventually as far as Hawaii, Tahiti, and New Zealand, carried by the ancestors of what linguists call the Austronesian language family, one of the largest and most geographically dispersed language families on Earth. When geneticists compared modern Taiwanese indigenous populations, whose ancestors are believed to represent an early branch of this expansion, against ancient farming populations recovered from mainland China, the genetic bridge connecting them was unmistakable. Direct physical proof that a population expansion beginning somewhere along China's southeastern coast several thousand years ago ultimately seeded human settlement across roughly half the surface area of the Pacific Ocean, one of the most extraordinary maritime migrations in human history, and one whose true scale was only fully confirmed once ancient and modern DNA could be compared side by side. Meanwhile, in China's far west and southwest, a completely different and in some ways even more remarkable genetic story was unfolding on the roof of the world. The Tibetan Plateau, sitting at an average elevation above 4,000 meters, presents one of the most physiologically punishing environments any human population has ever successfully and permanently settled, with oxygen levels so thin that lowland visitors frequently suffer severe altitude sickness within hours of arrival. When researchers sequenced the genomes of Tibetan populations and compared them against lowland Han Chinese populations, they discovered that Tibetans carry a specific genetic variant of a gene called EPAS1, at dramatically higher frequency, a variant that allows their bodies to use oxygen far more efficiently at high altitude without the dangerously thickened blood that plagues lowland populations attempting to adapt to similar elevations. This alone was a fascinating example of rapid human evolution in action. But then came the twist that stunned the entire genetics community. When researchers traced the origin of this specific EPAS1 variant, comparing it against the genomes of Neanderthals, modern humans worldwide, and critically, against the genome of the mysterious extinct human relatives known as Denisovans, first identified from a fingerbone fragment in a Siberian cave. They discovered that the high-altitude adaptation carried by millions of Tibetans today did not evolve independently within modern humans at all. It was inherited directly from interbreeding with Denisovans, tens of thousands of years ago, a genetic gift passed from an entirely separate, long-extinct branch of the human family tree, quietly preserved and actively selected for within a single modern population because it offered a life-or-death survival advantage in one of the most extreme environments on Earth. It stands today as one of the most vivid, concrete examples ever documented of archaic human DNA directly shaping the survival and biology of people alive right now. As researchers expanded their comparative net outward from China's borders, connecting Chinese genomes against samples collected from Japan, Korea, Mongolia, Southeast Asia, and eventually across the Bering Strait into the Americas, an enormous, sprawling web of ancient relationships began to emerge. Comparative studies examining Japanese population genetics revealed that the modern Japanese gene pool represents a layered blend of at least two major ancestral populations, an earlier hunter-gatherer population known as the Jomon, whose genetic signature closely resembles even older lineages found scattered across the East Asian mainland, and a later wave of rice farming migrants who arrived from the Korean peninsula and, more distantly, from mainland China, beginning roughly 2,500 years ago, bringing wet rice agriculture and metalworking technology with them. Korean populations show their own distinct but related genetic layering, connected both to the same ancient East Asian mainland ancestry and to additional influences from further north in Siberia and Manchuria. And when geneticists turned their attention across the Pacific entirely, comparing ancient Siberian genomes, themselves closely related to ancestral populations found in northern China, against the genomes of indigenous peoples throughout North and South America, they confirmed what archaeologists had long suspected but could never definitively prove using stone tools and pottery shards alone, that the ancestors of virtually every indigenous population across the entire Western Hemisphere descend from a relatively small founding population that crossed from Siberia into Alaska via the Bering Land Bridge sometime between 15 and 23,000 years ago, a population whose own deepest genetic roots trace directly back through ancient Siberia into the broader family of East Asian lineages that also gave rise to the ancestors of modern China. This means that, in a very real, scientifically documented sense, the genetic threads connecting a farmer in rural Sichuan province to an indigenous community in the Andes Mountains of South America, thousands of miles and thousands of years apart, are more directly traceable and more recently shared than the genetic threads connecting that same Sichuan farmer to certain populations in Western Europe or Sub-Saharan Africa. Geography, climate, and the specific timing of ancient migrations, far more than any concept of fixed racial category, are what actually shaped and continue to shape the patterns of human genetic relatedness visible across the modern world. The comparative research did not stop at tracing ancient migration routes. Scientists also turned their attention to a more recent and, in some ways, more socially consequential chapter of Chinese genetic history, the demographic expansion of the Han Chinese population itself. Roughly 2,000 years ago, during the era of the Han population growth, combined to drive what geneticists now recognize as one of the most significant demographic expansions in population growth, combined to drive what geneticists now recognize as one of the most significant demographic expansions in population growth, combined to drive what geneticists now recognize as one of the most significant demographic expansions in population growth, combined to drive what geneticists now recognize as one of the most significant demographic expansions in recorded human history. Comparative genetic analysis reveals a clear, measurable signature of this expansion, an event in which populations originally concentrated in the Yellow River Valley of northern China spread outward over the following 2 millennia, gradually absorbing, intermarrying with, and genetically blending alongside numerous distinct indigenous populations already living across what is now southern and southwestern China, including groups whose more distant ancestry connects to the same ancient southern coastal migration route that populated much of Southeast Asia. This is precisely why, even today, when researchers compare Han Chinese genomes sampled from different regions of the country, they find measurable, mappable genetic differences correlating closely with geography, reflecting this long, gradual, multi-thousand-year story of expansion and absorption rather than any single uniform ancestral population arriving all at once. It is a genetic portrait, not of a monolith, but of one of the largest and longest-running processes of population blending in all of recorded human history. There is also a smaller, quieter, but no less remarkable thread running through this research. the genetic history of China's numerous officially recognized ethnic minority groups, from the Uyghur populations of China's northwest, whose genomes reveal a fascinating and historically significant blend of East Asian and West Eurasian ancestry, reflecting the region's ancient position along the Silk Road, the network of trade routes that for centuries carried goods, religions, ideas, and inevitably genes between East Asia, Central Asia, Persia, and the Mediterranean world, to the Zhuang, Yi, Miao, and dozens of other officially recognized minority populations scattered across southern and southwestern China, many of whom carry genetic signatures connecting them closely to the earliest ancient populations of the region, communities whose ancestry in some cases appears to have remained relatively continuous and distinct across thousands of years, even as the broader Han Chinese population expanded and absorbed many neighboring groups around them. Comparative genome sequencing of these minority populations has proven scientifically invaluable precisely because it offers researchers a series of relatively less mixed genetic windows into China's deep ancestral past, helping to calibrate and cross-check the broader migration timeline reconstructed from ancient DNA and from the more heavily blended genomes of the majority population. Perhaps the single most quietly profound finding to emerge from this entire body of comparative research, spanning ancient cave skeletons, modern population surveys, and everything in between, is simply this. Every attempt to draw a clean, objective, biologically meaningful line around a Chinese race, an Asian race, or indeed any race at all, dissolves the moment researchers actually look closely enough at the genome. And what exists instead, in China as in every other region of the world scientists have studied with comparable rigor, is an almost unimaginably intricate, continuously overlapping tapestry of migration, adaptation, isolation, and reunion, stretching back tens of thousands of years, shaped far more powerfully by geography, climate, agriculture, and the simple universal human tendency to eventually meet, trade with, and marry one's neighbors than by any fixed biological boundary. The old racial categories that once dominated scientific thinking were, ultimately, crude approximations built from surface-level observations, skin tone, eye shape, hair texture, superficial traits shaped by relatively recent and relatively superficial evolutionary pressures like sun exposure and climate adaptation, layered on top of a far deeper, far more complex, and far more interconnected genetic history that those simple observations were never capable of capturing. None of this diminishes the real, rich, and deeply meaningful cultural, linguistic, and historical identities that exist across China and the wider world today. Ethnicity, language, culture, and shared history remain profoundly real and important dimensions of human identity and belonging, shaped by lived experience, tradition, and community in ways that genetics alone can never fully capture or explain. What the science does do, decisively and repeatedly, is strip away the false biological authority that outdated racial categories once claimed for themselves, revealing that the deep genetic story of humanity in China and everywhere else on Earth was never one of separate, static, biologically bounded races, quietly evolving in isolation from one another. It was, and remains, one single, continuous, magnificently tangled story of a species that has never truly stopped moving, never truly stopped mixing, and never truly stopped becoming something new. Somewhere in a research freezer today, another ancient bone fragment sits waiting its turn, another chapter of that story waiting patiently to be read, and with every new genome sequenced, the picture grows not more divided, but more unified, more intricate, and more astonishing than the generations before us ever could have imagined.